Okay. Can everyone hear me? Okay, so we'll begin, yeah? Okay. Now, today's lecture is going to be about 3 lectures, but the content of it is about like 7 or 8 lectures, which are included in your MOL 2 final. So hopefully we can use this opportunity to review a lot of material for you guys. But this is my own presentation, by the way. I looked at Dr. Hana's lectures. I sort of summarized them according to what I felt was appropriate. And it's about 30 slides. If you want, I can send it to you later as well. Okay. So this first slide is showing you why you eat, essentially. You eat because in a meal you have carbohydrates, proteins, and fat. Carbohydrates are broken down into glucose. Glucose is converted through glycolysis to pyruvate. Pyruvate is converted to acetyl-CoA by acetyl-CoA. Proteins as well. Proteins, when you absorb them, those amino acids go to the liver, and amino acids are basically a carbon skeleton with an amino group attached to it, right? So you remove the amino group, and now you just have the carbon skeleton. That carbon skeleton can be used to make different things. One of those things is pyruvate, which goes into acetyl-CoA. Fats as well, you break down fatty acids by a process of beta oxidation to acetyl-CoA. So notice when you're using up energy, all of your nutrients are being funneled down to acetyl-CoA. That acetyl-CoA is going through the Krebs cycle to generate NADH and FADH2. These carry high energy electrons. These electrons then on the inner mitochondrial membrane pass through the electron transport chain, and you capture some of that energy to make ATP. The rest of the energy is used to maintain body temperature. So that's just an overview of what energy usage looks like. But then we have this curve. This is a glucose tolerance curve, and this is important for your understanding, okay? So the x-axis shows you time, the y-axis shows you blood glucose. So changes in blood glucose over time. At this point, you eat a meal, blood glucose goes up, and then eventually within 40 minutes, blood glucose comes back down. Yeah. So can anyone tell me what hormone is activated here? What hormone is causing this decrease in blood glucose? Insulin, exactly. So insulin, if you understand this concept, it's very easy to understand that insulin activates pathways which use up blood glucose or store blood glucose. Okay, and we'll look at those pathways later. But then it's easy to understand as well that if you don't have insulin, like in diabetes, blood glucose remains high, so you have hyperglycemia. But notice in diabetes, blood glucose does also come down. Why does blood glucose come down if there's no insulin? Does anyone know? In diabetes, you don't have insulin, right? You have a lack of insulin effect, either because you don't have insulin like in type 1, or you have insulin, but the cells are not responsive to insulin like in type 2. But notice that in diabetes, blood glucose still comes down without insulin. So why does blood glucose come down without insulin? It's being excreted. And normally, your kidneys reabsorb all of the glucose. So actually what happens is there are cells in the body which don't rely on insulin, which don't use insulin. And they're basically minding their own business, and they're taking up glucose all the time at a steady pace. When they take up glucose, blood glucose slowly goes down. The 2 main cells you need to know are the red blood cell and the brain. These 2 tissues they don't rely on insulin for taking up glucose and using glucose. So in diabetes, this decrease in blood glucose is because of insulin-independent cells. From this slide, I just need you to know this, that diabetes blood glucose comes down because there are insulin-independent cells, namely red blood cells and the brain. So now, this is what Dr. Hana said. I just summarized it. Now in metabolism, you have many different pathways, right? So you have gluconeogenesis, glycolysis, fatty acid synthesis, fatty acid breakdown. And a lot of people have trouble sort of understanding how to fit all of this in together. You need to ask 4 basic questions if you're going to understand metabolism. Firstly, what condition is the body in? i.e. if is it well fed, or is there high blood glucose, or is the body starving or fasting, or if there's low blood glucose. So let me ask you guys answer these questions for me. If the body is well fed, what hormone will the body release? If the body is in a well fed state, it will release insulin. If it's in a fasting state, it will release glucagon. So the condition that the body is in, or the nutritional status of the body, tells you which hormone the body is releasing. This determines which pathways the body has activated. So we said that insulin likes to lower blood glucose, right? So insulin will lower blood glucose by stimulating pathways which either use up the glucose, like glycolysis, or they store the glucose, like glycogen synthesis or fatty acid synthesis. Fatty acids then go to the adipose tissue to be stored. So all of these pathways will be activated by insulin. All of the other pathways will be activated by glucagon. So fatty acid breakdown, glucose production, because glucagon is activated in a starvation condition. So it will want to produce glucose. How does it do that? It inhibits glycolysis, it inhibits fatty acid synthesis, it inhibits glycogen synthesis, and it causes glycogen breakdown, it causes gluconeogenesis, and it causes fatty acid breakdown. So whenever you get a question in biochemistry about metabolism, first try to figure out what condition the body is in, because a lot of questions start with the guy just ate a high carbohydrate meal. If the guy just ate a high carbohydrate meal, that means the body has high blood glucose. That means the body is releasing insulin. That means that from that you can know which pathways are activated. The next question is, what are the mechanisms by which insulin or glucagon regulate these pathways? There are two main pathways you need to know. The third one is minor. So the first one is gene expression. So insulin binds to its receptor on the cell membrane, which is a tyrosine kinase class of receptor. That downstream activates a signaling cascade, which activates a phosphatase, and it induces gene expression. It should make sense to you that gene expression takes a long time, because in gene expression, the RNA polymerase has to go inside the nucleus. It has to bind to mRNA. It has to bind to the promoter of the gene. It has to transcribe the mRNA. The mRNA has to be capped at the 5' end. It has to be polyadenylated. It has to be spliced. It has to go out into the cytoplasm. It has to be translated. Then it has to be modified, and then you have the final protein. So gene expression is slow to occur. The pathway which is quick to occur is covalent modification. Covalent modification refers to phosphorylation and dephosphorylation. What you need to know is insulin causes dephosphorylation. Glucagon causes phosphorylation. So if you understood these concepts, I'll repeat them one more time and then I'll ask you guys. If the body is in a well-fed state or is in a starved state, that's the first question. That will determine which hormone is activated, which is the second question. Which hormone is activated will determine which pathways the body stimulates, whether it uses up glucose or it produces glucose. And then you need to know that insulin and glucagon regulate these pathways by gene expression, which is slow, and by covalent modification, by phosphorylation, dephosphorylation. Insulin causes dephosphorylation, glucagon causes phosphorylation. So now if you understood these 4 questions, you should understand regulation. So let me ask you, fatty acid synthesis, let me give you an example. Fatty acid synthesis, in which nutritional status must the body be in for you to activate fatty acid synthesis? Does fatty acid synthesis occur in a well-fed state or the starvation condition? Well-fed, because that's when your body wants to store that energy. That means fatty acid synthesis, that means if it's well-fed, that means insulin will be activated. That means fatty acid synthesis will be activated by insulin. Insulin works through 2 mechanisms we discussed, gene expression and dephosphorylation. That means all of the enzymes in which fatty acid synthesis is active are activated by dephosphorylation. Does that make sense to everyone? So if you can understand this concept, you can understand all of regulation, okay? Basically, this is just a summary slide of what we discussed. Insulin is pulling the body's metabolism in a direction of glucose or energy storage. So it's decreasing glycogen breakdown, it's decreasing glycogen production. glucose production is decreasing fatty acid breakdown. Glucagon and epinephrine are pushing the body in a pathway of energy production. So they're producing glucose, they're breaking down fatty acids. Which of these pathways is activated depends on the nutritional status of the body. If the body has high blood glucose, the body releases insulin, suppresses glucagon. If the body has low blood glucose, the body releases glucagon and epinephrine, suppresses insulin. So in a low blood glucose, these pathways are activated. In a high blood glucose, these pathways are inhibited. These are just general concepts. You should remember them because at the end, I'll try and tie in everything together. Now, let's look at a few examples of some pathways, okay? This is glycolysis. You took this in MOL 1, so I won't go into much detail here. But what you need to know is that glycolysis asks these questions again. What condition must the body be in for you to stimulate glycolysis? Well-fed or starvation? Well-fed, because it's using up glucose, right? So that means the body must be releasing insulin. That means insulin must activate glycolysis either by gene expression or by phosphorylation. So what happens when you eat a high carbohydrate meal, glucose comes into the cell. Every cell has an enzyme called hexokinase. Hexokinase phosphorylates glucose, which traps the glucose in. In the liver, and in the liver only, you have an enzyme called glucokinase. Glucokinase basically has a capacity to deal with all of that extra glucose that's coming in. So insulin activates glucokinase, which should make sense because insulin is activated in this condition. Insulin activates glucokinase by which mechanisms? Either dephosphorylation or gene level. Insulin activates glucokinase at the gene level. The next step I want you guys to remember is this one, PFK1. This is basically the rate limiting step of glycolysis. This is also activated by insulin, and insulin activates an enzyme called PFK2 by dephosphorylation, which should make sense again. And this way glycolysis acts. So glycolysis occurs when you're well-fed. That means the body is releasing insulin. That means insulin activates glycolysis. How does insulin activate glycolysis? It activates glucokinase at the gene level, and it activates PFK2 by dephosphorylation. That's all I need you guys to remember. The next pathway is glycogen synthesis. All I need you guys to know is glucose. Then answer these questions for glycogen synthesis. In what condition must the body be in for you to synthesize glycogen? Well-fed or starvation? Well-fed again? No, well-fed, because glycogen, you're storing glucose. You would store glucose when you have a lot of glucose. That means you're well-fed. So the glycogen synthesis is occurring when you're in well-fed conditions. That means which hormone is the body releasing? Insulin. That means insulin will activate glycogen production. How will it do this? Glycogen is produced by an enzyme called glycogen synthase. Insulin activates this enzyme by which mechanism? Dephosphorylation. Conversely, glycogen breakdown or glycogenolysis is occurring by an enzyme called glycogen phosphorylase. Obviously, glycogen breakdown would occur when you have low blood glucose. That means low blood glucose activates glucagon. That means glucagon is activating glycogen breakdown. By which mechanism? Glucagon phosphorylates glycogen phosphorylase. And that causes glycogen breakdown. That's all I need to remember. I hope these pathways are now making sense. This is a review. That's why I included most of these pathways, because these are in your pre-midterm lectures. So I thought this would be a good opportunity for you guys to review this. Now, let me ask you a question. And this is interesting. Your body stores energy either as glycogen or as fatty acid or as fat. Which is the major storage form? Is it glycogen or is it fat? Does your body store more glycogen or more fat? What is the major storage in the body? One guy says fat. Yara, fat. Okay. Now let me ask you why though. You're right, it is fat. Glycogen stores are actually very low. Why is fat the major storage form in the body? Because glycogen, if you remember from your MOL 1, the structure of glucose, glucose has those oxygen molecules and carbon molecules. That means glucose is osmotically active. That means glucose likes to attract water. So if you synthesize a lot of glycogen, exceeding the capacity, that will attract water and the liver will swell up. Glycogen is mainly stored in the liver, right? So if you store a lot of glycogen, because glycogen is osmotic, it will attract a lot of water and the liver will swell up. That's why, for example, in glycogen storage diseases, which you did in MOL 1, the main problem is that there's too much glycogen in the blood, too much glycogen in the liver, and that causes hepatomegaly. So that's why glycogen, you can only store a little bit of that. Fat, on the other hand, is hydrophobic. You can store as much of it as you like because it doesn't attract water. So what does the body do? And when you eat a high carbohydrate meal, is that clear? Yeah, good. When you eat a high carbohydrate meal, the body initially stores glycogen. So it needs to replenish the glycogen stores. But eventually you can't synthesize more glycogen. So what do you do with that extra glucose? You make it into fatty acid. So let me show you how. Glucose enters the cell. We're talking about the liver here. Glucose is converted to acetyl-CoA. This is all glycolysis, and this is converted to citrate. Citrate will accumulate inside the cell, and citrate will go back out into the cytoplasm. Citrate runs into an enzyme called citrate lyase, which generates oxaloacetate and acetyl-CoA. Oxaloacetate goes back into pyruvate by the malic enzyme, which generates NADPH. Pyruvate goes back in to the mitochondria. What I want you to remember for the sake of this lecture is this step, acetyl-CoA. Acetyl-CoA gets converted to malonyl-CoA by an enzyme called acetyl-CoA carboxylase. And this enzyme requires ATP, biotin, and carbon dioxide. So it's an ABC carboxylase. Now, malonyl-CoA generates fatty acid. Those two steps are the most important for you to know. Now, going off our understanding of regulation, we said fatty acid synthesis occurs in well-fed conditions. That means fatty acid synthesis will be activated by insulin. And insulin will activate acetyl-CoA carboxylase, which should make sense now. And it also activates fatty acid synthase. And Dr. Abdul Jabbar likes to ask about this. He says fatty acid synthase has 4 steps by which it synthesizes the fatty acid: oxidation, reduction, dehydration, reduction. Okay, so again, glucose is initially stored as glycogen. When your glycogen stores are full, glucose is made into fatty acid. What the steps I need you to know is acetyl-CoA to malonyl-CoA, malonyl-CoA to fatty acid. Acetyl-CoA, this is the important step, acetyl-CoA carboxylase, activated by insulin, which should make sense. And then fatty acid synthase, which is also activated by insulin. These fatty acids go to the endoplasmic reticulum of the liver cell, and they're joined to the glycerol molecule. This forms a triglyceride. Then this triglyceride is packaged in VLDLs, and those are sent into the bloodstream. Does this pathway make sense to you? All of this? Yes? Anyone else? Yeah, good. So from this pathway, I need you to remember this, and I need you to remember that fatty acids are made into triglycerides, packaged in VLDLs, and then sent out into the bloodstream. Now, this is a long pathway, but we will try our best to summarize it. This deals with lipoprotein metabolism. Lipoproteins are basically lipids with a protein attached to them, and inside the lipoproteins, you have fat stored. And there are basically taxis which transport the fat around the body. Before we get into this, I need you guys to remember some apoproteins. The first apoprotein is ApoA. I need you to remember that it's in HDL and it activates LCAT. We'll get to the function of LCAT later. The next apoprotein is ApoB. There's 2 types, B100, which is found in VLDL, IDL, LDL, and B48, which is found in chylomicrons. The next protein is ApoC2, and I need you to know it activates lipoprotein lipase. The last apoprotein is ApoE, and I need you to know that it stimulates uptake of these lipoproteins into the liver. I need you to know these 4 functions. Now, let's say you just ate a meal containing fat. That fat is going to get broken down by the intestine through the action of bile and pancreatic lipase. That fat is going to get absorbed into the enterocyte, the intestinal cell, and it's going to get remade into triglycerides and cholesterol esters. Those remade triglycerides are going to get packaged in chylomicrons. Those chylomicrons require ApoB48. The chylomicrons through ApoB48 are secreted into the lacteal, into the lymphatics. Remember that fats are absorbed into the lymphatics, right? That's because of chylomicrons. Chylomicrons have ApoB48 on them. Eventually, chylomicrons travel through the thoracic duct and they end up into the bloodstream. In the bloodstream, they bump into another lipoprotein called HDL. HDL gives chylomicrons ApoE and ApoC2. So now the chylomicrons have three apoprotein, B48, which was originally there, ApoE and ApoC2, it got from HDL. What happens? As the chylomicron travels through the circulation, ApoC2 activates an enzyme called lipoprotein lipase. Lipoprotein lipase is on the endothelium, is on the capillaries of the endothelium. Okay, so you have capillaries in the adipocyte. On that capillary is lined by endothelium. On the surface of that endothelium, you have an enzyme called lipoprotein lipase. That lipoprotein lipase is activated by ApoC2. It basically extracts the triglyceride, hydrolyzes it to form fatty acid, and that fatty acid is stored in the adipose tissue and in the muscle. Eventually, because these triglycerides are being extracted from chylomicrons, the chylomicrons are shrinking. Then they become chylomicron remnants. And notice now they don't have the triglyceride anymore, as much triglyceride anymore, because they've given it off. So they donate ApoC2 back to HDL. Now it's back with HDL. And these chylomicron remnants are taken up by the liver through the action of ApoE. Okay, so B48 required for exocytosis into the lymphatics originally. ApoC2 and ApoE they get from HDL. ApoC2 activates lipoprotein lipase. Lipoprotein lipase extracts these fatty acids and stores them in the adipose tissue and muscle. As a result of this extraction, the chylomicron shrinks to form chylomicron remnants. These chylomicron remnants give back ApoC2 to HDL, and they're taken up into the liver by ApoE. Now, at the same time that chylomicron remnants are being taken up by the liver, from the last slide, you should know that fatty acids are being synthesized by the liver, and they're being joined to glycerol to form triglycerides. Those triglycerides are then being packaged in VLDLs. Some of that cholesterol and triglyceride, which is coming through chylomicron remnants, is then also packaged in those same VLDLs, and they then are released into the bloodstream. VLDLs contain ApoB100, and then it's the same story as with chylomicrons. They bump into HDL. HDL gives them ApoE and ApoC2. I forgot why they need C2. C2 activates lipoprotein lipase here. This is the function you have to remember. ApoC2 activates lipoprotein lipase. So VLDL originally has only B100. It bumps into HDL. HDL gives it ApoE and ApoC2. C2 activates lipoprotein lipase, which extracts these triglycerides from the VLDL into the adipose tissue and in the muscle. As a result, the VLDL sort of shrinks in size, and now it forms IDL. IDL, because it's given off that triglyceride, it gives off C2 back to the HDL. Because look, so it doesn't have C2 anymore. VLDL had C2, IDL gives off that C2 back. Now the IDL has two options. It can either go back into the liver by the action of ApoE. ApoE causes uptake by the liver, or it can run into an enzyme on the liver called hepatic lipase. Hepatic lipase also extracts any more triglycerides from the IDL. As a result, the IDL shrinks even more. Now that IDL gives off ApoE back to HDL as well. Now IDL shrunk plus gave off ApoE back to HDL. Now it is called an LDL. LDL only contains B100, so it doesn't contain ApoE. LDL, the problem is that cells express LDL receptor. Most of the LDL is going to get taken up by the liver, but the problem is some of that LDL is also going to go out into the periphery because other cells express LDL receptors. Now, what's going to happen? LDL by itself is not really a bad thing. You guys may hear that LDL cholesterol is bad cholesterol. What can happen is this B100 can get oxidized. by reactive oxygen species. This LDL will become oxidized LDL. Oxidized LDL is recognized by macrophages as being foreign, i.e. they don't belong in the body. So macrophages take up these LDLs, and LDLs are very high in cholesterol. So by taking up these LDL, the macrophages accumulate cholesterol. These macrophages are now called foam cells. Eventually, these macrophages will burst, releasing all of that cholesterol into the vasculature, and this will cause formation of atherosclerosis. This is where HDL comes in. HDL has ApoA, right? ApoA activates an enzyme called LCAT. LCAT takes that cholesterol in the periphery, stores it in HDL, and HDL goes back to the liver. So HDL transports cholesterol from the periphery to the liver. LDL transports cholesterol from the liver to the periphery. That's why HDL is called good cholesterol. Okay. What I need you to know right now, and I hope this pathway made sense, but what I need you to know and focus on is that LPL is activated at the gene level by insulin, which should make sense because after a meal, you have insulin, right? So insulin is activating LPL at the gene level. Now, let's move on to gluconeogenesis. Not much you need to know here. Just remember that gluconeogenesis requires energy. Okay, gluconeogenesis requires energy. So you're using ATP and you're using GTP. Because gluconeogenesis requires energy, you need to get this energy from somewhere. Where do you get this energy from? You get this energy from beta oxidation of fatty acids. Okay, gluconeogenesis is energy requiring. Where do you get the energy from? From oxidation of fatty acids. The next step I need you to know is this one, PEPCK, PEP carboxykinase. This is activated at the gene level by glucagon. Okay, this is activated by the gene level by glucagon. These questions should make sense to you now. What condition must the body be in for you to carry out gluconeogenesis? You're producing glucose there. Therefore, what condition must the body be in? Does anyone know? It should be fasting. Therefore, body should be releasing glucagon. Therefore, glucagon must be activated by activating gluconeogenesis. Also, glycogen breakdown must be being activated at the same time, right? By which mechanism? Glycogen's breakdown was being activated by phosphorylation. Gluconeogenesis is being activated by the gene at the gene level. Does this all make sense to you now? Or am I going too fast? Yes? Good. Okay. Now, remember what I told you in the first slide, that gene expression takes a long time to occur, while phosphorylation, dephosphorylation occurs very quickly. So it doesn't make sense to you that because glucagon is regulating gluconeogenesis at the gene level, gluconeogenesis takes a bit of time to kick in. Glycogen breakdown is activated by the phosphorylation, so glycogen breakdown occurs immediately. Does that make sense? Yes? Okay, that's good. Because we look at a graph when you're fasting, glycogen stores are immediately broken down, whereas gluconeogenesis takes a bit of time to come up. And that's because of this mechanism, because gluconeogenesis is being activated at the gene level, while glycogen breakdown is being activated by phosphorylation, and gene expression takes time. Let's move on to lipolysis. I know we've covered a lot of material. Are you guys following, or should I repeat anything? Is it making sense so far? Yes? Okay, that's good. Now, lipolysis is fatty acid breakdown of triglycerides, right? So what condition must the body be in for you to break down triglycerides? Fasting or well-fed? Fasting, that's good. What hormone must the body be releasing when you're in fasting? Glucagon, yeah. So the body is releasing glucagon, but the body is also releasing epinephrine. Glucagon, what you should know is that glucagon acts on the liver only, while epinephrine acts on fats, adipocytes, and muscle cells. So the changes you see in the adipose tissue and in the muscle are not because of glucagon, they're because of epinephrine. The changes which you see in the liver are because of glucagon. So what happens here? in the adipose tissue here, triglycerides are broken down into glycerol and fatty acid by hormone-sensitive lipase. Contrast to the previous slide, we said lipoprotein lipase is activated by insulin, causes deposition of fat. Hormone-sensitive lipase breaks down triglycerides. Let me ask you guys a question. In which situation would hormone-sensitive lipase be active? Phosphorylation, dephosphorylation? Because you understood these questions, you should know in which situation should this enzyme be active, phosphorylation or dephosphorylation? Phosphorylation, right? Because glucagon and epinephrine are the ones being active. And I told you that they cause phosphorylation. Insulin causes dephosphorylation. So if these are activated by epinephrine and glucagon and cortisol, that means they should be activated by phosphorylation. That's good. Now the triglyceride is hydrolyzed by hormone-sensitive lipase or HSL to form glycerol and fatty acids. They have two very different functions, and you need to know that this will come in your exam, by the way. What the pathways I've discussed are high yield. They do come in exams, not just in MOL, but in step 1 as well. The glycerol goes into the liver. And if I can show you here, you notice here that glycerol is an intermediary of glycolysis. That means glycerol can be used in gluconeogenesis to just form glucose here. So the glycerol, which is coming from the from the adipose tissue, is coming into the liver, is being converted into glucose by gluconeogenesis. Okay, and we said also that gluconeogenesis is energy requiring. Where do you get this energy from? From beta oxidation of fatty acids. So in summary, triglycerides are being hydrolyzed into glycerol and fatty acid. Glycerol is being used by the liver to produce glucose. The fatty acids are being used by the liver to fuel gluconeogenesis. The fatty acids themselves are not being used to make glucose. They're being used to fuel gluconeogenesis because gluconeogenesis is energy requiring. Does that make sense? No? Should I repeat? Okay, I guess we can move on. So, because fatty acids generate okay, so the question is, why aren't fatty acids being used to make glucose? Glycerol is being used to make glucose. Why aren't fatty acids being used to make glucose? Because fatty acids generate acetyl-CoA. And acetyl-CoA, the carbons of acetyl-CoA cannot be used to make glucose. There's a whole concept to this, but essentially in the Krebs cycle, the carbons that are used to make acetyl-CoA are being removed. And none of those carbons which comprise acetyl-CoA can be used to make glucose. That's the general concept you need to know. So again, glycerol is being used to make glucose by gluconeogenesis. Fatty acids are being used to make acetyl-CoA, which can be used in the Krebs cycle to generate ATP. This ATP is going to be used to fuel gluconeogenesis. Some of that acetyl-CoA is going to get converted into ketone bodies. Now, this is very important for you guys to understand. Fatty acids are used as a fuel by the liver, by the muscle, skeletal and cardiac in starvation conditions for energy. So in normal conditions, these tissues utilize glucose. In starvations, they switch to fatty acids. However, fatty acids are because they're hydrophobic and they're bound to albumin, they cannot cross the blood-brain barrier. That means the brain cannot use fatty acids. That means in starvation condition, the body must find a way to supply energy to the brain. How does it do this? It makes these fatty acids into ketone bodies. Ketone bodies are soluble, so they can cross the blood-brain barrier. And in this way, the brain can utilize ketone bodies. Does that make sense? Yep. Okay, good. So again, you should know this pathway. You should know that it's activated in a starvation condition. It's activated by epinephrine, because I told you glucagon mainly acts in the liver. Fatty acids are being used to generate ketone bodies, which can be used as a fuel by the brain. Remember that brain and the red blood cell were two tissues we discussed at the beginning of the lecture that are insulin independent. The brain can utilize acetyl-CoA. But the red blood cell cannot, because the red blood cell doesn't have mitochondria. So the red blood cell can only utilize glucose. So by the brain switching to ketone bodies, it spares that glucose for the red blood cell. Does that make sense? That's why the brain switches to ketone bodies. That's why your body switches to ketone bodies. Because firstly, it wants to supply energy to the brain. Secondly, by the brain utilizing ketones, it sort of spares that the glucose for the red blood cell, which can only utilize glucose. So now, I hope now this figure makes more sense to you. We went through all of these pathways, which covered like 9 lectures. So red blood cells cannot, because if you remember, glycolysis occurs in the cytoplasm, Krebs cycle occurs in the mitochondria. Red blood cells don't have mitochondria. So red blood cells can only carry out glycolysis. So the red blood cells can only use glucose. The brain has a mitochondria, so it can use the Krebs cycle. It can use ketone bodies. Okay? So by the brain switching to ketone bodies, it's sparing that little bit of glucose you have for the red blood cell. We'll discuss this in more detail. But now I hope this pathway, this diagram that we discussed earlier makes more sense to you. Now you guys should understand what happens in diabetes. Because in diabetes, you lack insulin effect. If you lack insulin effect, you have increased lipolysis because insulin causes storage. Epinephrine and glucose and cortisol cause breakdown. So as a lack of insulin effect, you have hyperglycemia because the liver is producing glucose plus it isn't using and storing the glucose. You have lipolysis because of unopposed action of epinephrine and cortisol. And because you're generating fatty acids, those fatty acids are going to the liver and they can generate ketone bodies. So the result of diabetes, whether type 1 or type 2, is hyperglycemia, lipolysis, and ketogenesis. Does this make sense? Clear? Yeah. So it is just this diagram. Because of lack of insulin effect, will decrease glucose uptake by muscle and adipose, increase hepatic gluconeogenesis, increase lipolysis. So you have elevated free fatty acids, which can go to the liver to be generated in the ketone bodies. Plus you have hyperglycemia. Now, why is hyperglycemia bad for the body? How you can remember this for first year is just to remember that glucose loves to bind to proteins. Glucose loves proteins. It binds to proteins. One of those proteins, for example, could be hemoglobin, and this generates hemoglobin HbA1c. And you use this as a diagnosis of diabetes, elevated HbA1c. The next protein glucose can bind to is the basement membrane of the vasculature. Basically, what happens is when glucose binds to the basement membrane, that stimulates immune cells. Which causes inflammation, localized inflammation in that blood vessel. That causes the blood vessel to thicken. When the blood vessel starts to thicken, what happens to the lumen? It gets compressed. If the lumen gets narrow, you will have ischemia. If this blood vessel was a blood vessel of the leg, you will have ischemia of the foot, and diabetes is the most common cause of amputations. If you have this happening in the brain, you will result in a stroke, and diabetics are susceptible to strokes. If this happens in the coronary arteries, what will happen? MI, and diabetics do get higher incidence of MI. So does this pathogenesis of diabetes make sense? Yeah, okay. I hope you guys are finding this interesting because a lot of people don't really understand biochemistry. But if you understand biochemistry, it's a lot more fun to understand. Okay, so now this diagram is a bit scary, but it shows you all of what we've discussed happening at the same time. This figure shows you what happens in your body when you eat a high carbohydrate meal. When you eat a high carbohydrate meal, you're well fed. That means your body releases insulin. What are the effects of insulin? Is this diagram. We'll discuss them. First, let's look at the insulin-independent cells, the red blood cells and the brain cells. you have to know this, that the red blood cells and the brain cells have Glut1 and Glut3. Those are the glucose transporters present in the red blood cell and in the brain. And these are insulin independent. I also told you that the red blood cell lacks mitochondria, so it can only carry out glycolysis. The brain has mitochondria, so it can use acetyl-CoA, which is responsible for ketone bodies, as we'll see later on, right? These are insulin independent. Then let's move on to the liver. The liver has another glucose transporter, and you need to know this. They will ask you this in the exam. I remember they asked us about this, that Glut2 is present in the liver cell. So in the red blood cell and the brain, Glut1 and 3. In the liver, you have Glut2. Notice that Glut2 is not dependent on insulin. So insulin uptake into the liver doesn't rely on, glucose uptake into the liver doesn't rely on insulin. Insulin tells the liver what to do with the glucose, but it doesn't tell the liver to take up the glucose. Okay, so you need to be careful of that, because a lot of people get confused that the liver is insulin dependent. That's why therefore Glut2 is activated by insulin. It's not activated by insulin. Glut2 is there in the liver. Insulin is telling the liver what to do with the glucose. Okay. Now, there's an important concept that you guys should understand. That, let me explain it to you. Are you guys aware of what Km is? Km. Yes. It basically shows you affinity, right? So the lower the Km, the higher the affinity. The higher the Km, the lower the affinity. Let's draw this. This is blood glucose. And this is uptake, glucose uptake, okay? We're going to draw how these transporters are working. Let's draw Glut1 and Glut3 first. Let's say this is 80 and this is 120. So this is the normal blood glucose, right? I'm going to draw how the Glut1 and 3 transporters work, okay? They work like this. Notice that they have a very high affinity for glucose because they have a very low Km, right? This way, even if I'm in the range of normal blood glucose is entering Glut1 and 3. This is 1 and 3. Blood glucose is entering or being taken up by Glut1 and 3 at a very constant level. Also, you should know that even if I decrease blood glucose, as I'm doing here, does the uptake by Glut1 and 3 change? It doesn't, because it's still at Vmax. Also, if I increase blood glucose, does the uptake change? It doesn't, because I'm still operating at Vmax. So the red blood cells and the brain cells are taking up glucose at a very constant pace, because Glut1 and Glut3 have a very low Km, which means they have a very high affinity. Does that make sense? Excellent. And this should make sense to you because the red blood cell and the brain are crucial. So you want them to have a constant supply of glucose, right? Now I'm going to draw Glut2 kinetics, okay? So I'm going to draw how Glut2 takes up glucose with changing blood glucose concentrations. Glut2 works like this, okay? So now notice, in Glut2, when I increase, this is Glut2. When I increase blood glucose, what is happening to glucose uptake by Glut2? By this diagram, if I increase blood glucose, if I eat a high carbohydrate meal and I increase blood glucose, what is happening to glucose uptake by Glut2? It's increasing. Good. And if I decrease it, it's decreasing glucose uptake. That's important because when you eat a high carbohydrate meal, your glucose in the blood rises. That has little effect on Glut1 and Glut3 because they keep taking up that glucose at a very constant pace. Glut2, on the other hand, will increase its uptake. And that way, all of that extra glucose can be dealt with the liver, can be dealt with by the liver, and that can go into the liver. Does that make sense? Excellent. So it makes sense to one person or everyone else as well? Yeah. So this also happens in the pancreas. The pancreatic beta cells also have Glut2. And we'll come back to this and I'll ask you about this. So GLUT2 increases its ability to take up glucose according to the substrate glucose concentration. Why GLUT2 at the end is a flat line? Because every transporter at one point reaches maximum, right? Every transporter reaches a maximum uptake. Yeah. So again, remember GLUT1, GLUT3, red blood cells, brain. Remember GLUT2 in the liver can increase uptake when you eat a high carbohydrate meal. All of these up transporters are insulin independent. Now let's review what we've discussed for the past half an hour. Glucose, now insulin is active, right? Insulin activates glycogen synthase to cause glycogenesis, to store the glucose as glycogen. Insulin activates glucokinase at the gene level. It activates PFK2 by dephosphorylation to convert glucose into pyruvate, and then pyruvate can be used to generate ATP. At the same time, insulin activates HMG-CoA reductase at the gene level to stimulate acetyl-CoA production into cholesterol, to stimulate cholesterol production. When glycogen stores are replenished, insulin activates acetyl-CoA carboxylase and fatty acid synthase to make that excess acetyl-CoA into fatty acid. That fatty acid is joined to glycerol to make triglycerides. Those triglycerides are packaged in VLDLs, as we said. Those VLDLs are now sent out into the bloodstream. Insulin then goes to the adipose tissue and tells the adipose tissue, I've told the liver to synthesize triglycerides and VLDLs, and they'll be delivering fats to you. So upregulate lipoprotein lipase on the endothelial cells. Chylomicrons are obviously also present at this point. So VLDLs and chylomicrons, they deliver fatty acids to the adipocyte. Now you may ask that, well, the glycerol doesn't go into the adipocyte, importantly. The glycerol doesn't go into the adipocyte. The glycerol goes back into the liver. Now, there's a question that you guys should ask. Where does the adipocyte get glycerol from? Insulin has a solution for this as well. Insulin activates GLUT4 transporters on the membrane of the adipose tissue and the skeletal muscle. But let's focus here. Insulin activates GLUT4 expression on adipose tissue. This increases glucose uptake into the adipocyte. Insulin activates glycolysis in the adipocyte. A byproduct of glycolysis is glycerol phosphate, as we discussed. As a result, what will happen? That glycerol will join to fatty acids to make triglycerides, and now those triglycerides are stored in the fat, in the adipocyte. Insulin, at the same time, activates GLUT4 expression on the muscle cells. This increases glucose uptake into the muscle cells. Insulin activates glycogen synthase by dephosphorylation to cause glycogenesis, to store that glucose as glycogen. Insulin also activates amino acid uptake into the muscle cells and protein synthesis, so you can build muscle mass. So you notice how insulin is doing everything when you just ate a high carbohydrate meal. Does all of this make sense? This is a very complex figure, I know. But insulin is doing so much. Clear? Okay. Good. So now, so now it should make sense to you what happens in diabetes. In diabetes, there's no insulin. So the liver and the body just gets confused about what to do with the glucose. So glucose, when it comes into the liver, it can't be phosphorylated by glucokinase because glucokinase isn't present. So glucose just goes back out of the liver. So you have hyperglycemia. Second, there's no GLUT4 expression on adipocyte and muscle cells. So glucose doesn't go into skeletal muscles and adipocytes. Therefore, you have hyperglycemia. There's no lipoprotein lipase activation. So you have less deposition of fat. That's why diabetics tend to lose weight. Okay. So all of these effects, if you understand the basic biochemistry, you can understand the pathology of diabetes. Last point I would like to make is about about glucokinase. You remember the graph I showed you about GLUT2 and GLUT1 and 3? That GLUT2 increases uptake when there's high blood glucose and decreases uptake when there's low blood glucose. The same is with glucokinase. The same situation is with glucokinase. The glucokinase, the hexokinase is like GLUT1 and GLUT3. So it's always working at a steady pace. while glucokinase has increases its activity based on glucose concentration. That's important for later on when we discuss insulin synthesis, because Glut2 and glucokinase are both present as also in the pancreas. But nevertheless, this is that diagram. I hope this makes sense to you. This is basically all of what I've said in words. This is what Dr. Hannah wrote. Immediately after a meal, the blood glucose levels rise, i.e. you're well fed. That means insulin is released. Insulin targets the liver, the muscle, and the adipocyte, as we said. As insulin causes glycogen synthesis in the liver, but when the capacity for glycogen is full, then you make that extra glucose into triglycerides, and those triglycerides are packaged in VLDLs sent to the adipose tissue. Insulin at the same time activates lipoprotein lipase at the gene level to cause deposition of triglycerides and as fatty acids to hydrolyze fatty acids, to hydrolyze triglycerides into fatty acids and glycerol. Glycerol will go back into the liver. Fatty acids will go into the adipocyte. Insulin at the same time activates glucose uptake into adipose tissue by activating Glut4 expression. It also does this in the muscle. Okay. Yeah. Now Dr. Hannah tells you that two tissues, the brain and the red blood cells, are insulin independent, which I hope makes sense to you guys now. The brain derives energy from glucose in both well-fed and fasting. But in prolonged fasting, as I told you, the brain switches to ketone bodies, which spares the glucose for the red blood cell, because the red blood cell can only utilize glucose. Does all of this make sense so far? So these are the pathways which are stimulated when you eat a high carbohydrate meal. Yes, it's a bit heavy, I know, and a bit difficult, but you know. Okay, good. This is a summary from first aid about the functions of insulin. Insulin binds to insulin receptors, which I said are tyrosine kinase class of receptors. What are the effects of insulin? It induces glucose uptake. Which transporter induces glucose uptake? Is insulin dependent. Which GLUT transporter is insulin dependent? Can you remind me? Which glucose transporter is insulin dependent? Glut4. And where is it present? Glut1 to 3 are in red blood cells and brain. They are insulin independent. One's in the brain, one's in the muscle, and one's in the adipose tissue. Glut4, excellent. So Glut4 insulin stimulates increased glucose uptake into skeletal muscle and adipose tissue by Glut4 expression. Insulin increases glycogen synthesis and storage by activating glycogen synthesis by dephosphorylation. Insulin increases triglyceride synthesis by activating acetyl-CoA carboxylates and fatty acid synthesis, what I told you. Insulin, as we said, increases protein synthesis. Skip this one, sodium retention. Skip this one. This you should understand from renal. Insulin obviously will decrease glucagon release. Insulin-dependent transporters, this is important for you to know. It probably will be in your exam. All of this you need to know. Glut4 is found in adipose tissue and striated muscle, skeletal muscle, and the Glut4 is insulin-dependent. Insulin-independent transporters, Glut1, red blood cells and brain. Glut2 is in the beta cells of the pancreas, like I told you, and in the liver. Glut3 is in the brain, also in the red blood cells. Brain utilizes glucose for metabolism normally, but it switches to ketone bodies during starvation. On the other hand, red blood cells always utilize glucose because they lack mitochondria for aerobic metabolism. So we're done with well-fed state, or we're done with insulin. Alhamdulillah. Now, let's discuss what happens in the body when you're starving or when you're fasting. This is very easy to understand, hopefully, since we've discussed all of the pathways. Blood glucose decreases when you're fasting. This stimulates glucagon release. Where is the action of glucagon? Can anyone remind me? I told you that glucagon mainly acts on the liver. Excellent. Glucagon acts on the liver. How? It stimulates glycogen breakdown by activating glycogen phosphorylase by dephosphorylation. This arrow shows you that. Glucagon activates gluconeogenesis by activating PEP carboxykinase at the gene level. So this shows you gluconeogenesis. All of these will increase blood glucose, contribute to maintaining blood glucose. Now there's there's one more question we need to answer here. That where do you get the pyruvate from? Because I think it was Yara that asked a question about why fatty acids aren't used to make glucose. And I told you that acetyl-CoA can't be used to make pyruvate. So where do you get the pyruvate from? That's the next question. You get pyruvate from three sources. One source, as I told you, the red blood cell can only carry out glycolysis. The pyruvate that the red blood cell generates is converted into lactate. The lactate goes into the liver. The lactate is regenerated into pyruvate, which is used for gluconeogenesis. That's the first pathway of producing pyruvate for gluconeogenesis. The second source is protein breakdown. As you know, in epinephrine, in fasting conditions, activates muscle protein breakdown. All those amino acids that are broken, proteins are broken down in the muscle, releasing amino acids. All of those amino acids are converted into alanine. Alanine goes back to the liver, goes to the liver. Alanine's amino group, the NH2 part, is removed for the urea cycle, and alanine is converted to pyruvate. That is the second source for gluconeogenesis. Okay, so first source lactate, second source alanine coming from muscle breakdown. The third source is hormone sensitive lipase. Which substance activates hormone sensitive lipase? Can anyone remind me? Just to make sure you guys are paying attention. Hormone sensitive lipase is activated by epinephrine, because glucagon mainly acts on the liver. Epinephrine acts on the adipose tissue and in the muscle. Hormone sensitive lipase is activated, and also cortisol activates hormone sensitive lipase. Hormone sensitive lipase is activated, it degrades triglycerides into glycerol and fatty acids. The fatty acids, the glycerol goes back to the liver, and I told you that glycerol is an intermediate of glycolysis. Therefore, glycerol phosphate can be used to make glucose. So that is the third way by which you generate that is the third source of gluconeogenesis. Glycerol coming from lipolysis gets converted to glycerol phosphate, that goes into gluconeogenesis. What happens to the fatty acid? The fatty acids are released into the bloodstream. They can be used by the muscle to make ATP, but importantly, they go to the liver. They get generated into they undergo beta oxidation, which is this step here, and they produce acetyl-CoA. Acetyl-CoA can be used to make ATP, and importantly, it's important for you to remember that this ATP fuels gluconeogenesis, like we discussed. The problem with fatty acids is they can't be used by the brain, because the brain fatty acids don't cross the blood-brain barrier. So how does your body solve this problem? It generates ketone bodies. Ketone bodies can be used by the muscle, but they can also cross the blood-brain barrier, as shown here, to generate acetyl-CoA. When acetyl-CoA is elevated in the brain, it inhibits this step, pyruvate to acetyl-CoA. This step is catalyzed by pyruvate dehydrogenase, remember? So this step is inhibited. Why is this important? Because this inhibits the utilization of glucose by the brain. Because you've inhibited this step, now the brain can no longer utilize glucose. What will that do? That will save the glucose for the red blood cell. So again, that's the whole diagram. First, look at this pathway. Glucose decreases, stimulates glucagon. Glucagon stimulates glycogen breakdown by activating glycogen phosphorylase. Glucagon also stimulates gluconeogenesis by activating PEP carboxykinase. Where do you get, the next question is, where do you get the pyruvate from? First source is lactic acid coming from the red blood cell. Second source is protein breakdown, which in the form of alanine goes to the liver to supply pyruvate. The third source is glycerol coming from fat breakdown. The fat goes into the liver to generate acetyl-CoA and ATP. ATP fuels glycolysis, but however, the fatty acids cannot be used by the brain because they don't cross the blood-brain barrier. So your body synthesizes ketone bodies which do cross the blood-brain barrier and are used by the brain as a fuel, and this will spare that glucose for the red blood cell. Look at this diagram here, this bottom right, and on the bottom right corner. It shows you that when you're fasting, glucose goes down immediately, but look, glycogen is immediately activated and it runs out within 24 hours. Glucagon is slow to kick in. Why is glucagon slow to kick in? Why does glucagon take time to occur? Because it's activated by gene expression, exactly. Glycogen synthesis is activated by phosphorylation, dephosphorylation, which is immediate. Like gluconeogenesis, glucagon activates PEP carboxykinase at the gene level, which takes time. Excellent. This is basically, again, summarized in words. Glucagon and epinephrine levels rise when you're fasting. These hormones exert their effects on skeletal muscle, adipose tissue, and the liver. Glucagon mainly on the liver, epinephrine on the skeletal muscle and the adipose tissue. In the liver, glucagon causes glycogen breakdown and gluconeogenesis. Although gluconeogenesis is slower than glycogen breakdown. The release of amino acids from the skeletal muscle causes formation of alanine. The alanine goes to the liver to supply pyruvate, which is used in gluconeogenesis. Fatty acids are liberated from adipose tissue. Also, they are taken up by the liver, and fatty acids provide the ATP necessary for gluconeogenesis. So again, this is summarizing what I've just told you. The release of amino acids and the release of fatty acids are stimulated by epinephrine, not glucagon. Glucagon acts on the liver only. Okay. Now, what happens if the fast is prolonged? Glucagon and epinephrine again are elevated. But you're generating excess acetyl-CoA, but because you're breaking down a lot of fat, because you've been fasting for a long time, your body is breaking down a lot of fat. So lipolysis is occurring rapidly. So you have a lot of acetyl-CoA. A lot of acetyl-CoA causes formation of ketone bodies. Ketone bodies are only synthesized by the liver. But let me ask you a question. Can the liver use ketone bodies? So liver is synthesizing ketone bodies, but it can't use ketone bodies. Why not? No? Yes? Okay. Good. So the liver can't use ketone bodies because it lacks an enzyme called thiopherase. Thiopherase is an enzyme which allows you to utilize ketone bodies. The liver doesn't have this enzyme. So the liver cannot use ketone bodies. Now, as I told you, the red blood cells, this sentence is important. The red blood cells have no mitochondria, so they continue to be dependent on glucose for their energy. I hope that all of this now makes sense. The brain adapts to using ketones for some of its energy after several weeks of fasting. The shift from glucose to ketones spares the red blood cell, spares the glucose for the red blood cell. Also, because you now don't need as much glucose, this spares the protein breakdown because you're breaking down a lot of protein, right? Because you're generating a lot of amino acids, which generates alanine for gluconeogenesis. So in fasting, you're generating a lot of protein breakdown, which gives rise to muscle wasting. This shift in the brain's metabolism from glucose to ketones will also spare some of that muscle breakdown. Now, this is the last slide of this lecture. The liver, as you know, utilizes glucose and amino acids in the well-fed state. So let's go back to this diagram. When you eat a high meal in proteins, amino acids go into the blood. As I told you, amino acids have an amino group and a carbon skeleton. The amino group is removed. The carbon skeleton is used to make pyruvate, which generates ATP. So that's what this sentence means. The liver utilizes glucose and amino acids for energy in a well-fed state. The liver utilizes fatty acids during fasting. It doesn't utilize ketones, as you guys said, because it lacks the enzyme thiopherase. The skeletal muscle in the well-fed state utilizes glucose. In the starvation condition, utilizes fatty acids and also ketone bodies. Cardiac muscle, you should know that they always utilize fatty acids and ketones in prolonged starvation. The brain, this is the most important part of this table. The brain utilizes glucose, but it utilizes ketone bodies in a prolonged fast. The red blood cell always uses glucose. I hope all of this makes sense now. I think, so we're done with this lecture. So any questions about this lecture? Clear. So I hope metabolism makes a lot more sense to you now. Nevertheless, I think Maghrib time. Yeah, now is Maghrib. So we'll take how about we take a break for Maghrib, 5 to 10 minutes, then we're gonna come back and finish the other two lectures. They shouldn't take that long. Okay? Is everyone okay with this? Yeah. So we'll take a break for 5 to 10 minutes for Maghrib. Now, this lecture we've discussed in the previous lecture the metabolic effects of insulin and glucagon. This lecture deals with the synthesis of insulin and the synthesis of glucagon, and basically their stimuli, what stimulates their release, what inhibits their release, these sorts of things. So it's not as high yield a lecture as the other one. That one I would say is one of the most important lectures for metabolism. This lecture is a bit, you can just remember a few points and I think you're going to be good. Okay, insulin, the structure of insulin, there's two chains, A and B. They're connected by two bonds, two disulfide bonds. That's all you need to remember. For synthesis, people concentrate too much on this. For me, I just remember protein trafficking. I think it was it was given to you guys in MOL 1. If you remember, proteins that are excreted from the body, not from the body, from the cell, or secreted from the cell, they have to go through a specific pathway. They are synthesized with a signal sequence at their end terminal. Then, as they're synthesized, they go to the endoplasmic reticulum. That signal sequence is removed. In the endoplasmic reticulum, then they go to the Golgi apparatus. Then another part of the protein is removed and they're packaged in vesicles. Then they're secreted from those vesicles by exocytosis from the cell. And this was the case for example, PTH, you guys did an MSK, it's synthesized as pre-pro-PTH. Pre is the signal sequence part. The pre-signal sequence is removed, so it becomes pro-PTH. And then another part is removed and it becomes PTH. Insulin is secreted. That means it has the same trafficking. Insulin is synthesized as pre-pro-insulin with a signal sequence. It moves into the ER where the signal sequence is removed to form pro-insulin. In the Golgi, then it goes to the Golgi apparatus, and that's where the pro part is cleaved. The pro part of insulin is stored with insulin in those vesicles, and that pro part is called C-peptide. Okay, so C-peptide is the pro part of pro-insulin. It's removed from insulin and is stored within the same vesicles, and their insulin and C-peptide are released together. Why is that important? Because you can measure C-peptide in the blood, and that gives you an idea of how good the beta cells are functioning. So let me give you an example. A guy presents to your clinic with hypoglycemia. You suspect there could be two situations here. There could be many situations, but for the sake of this example, there could be two situations. The first one is that the guy is hypoglycemic because he has a tumor or a cancer secreting insulin. He has a tumor secreting insulin. The second possibility is that he's injecting himself with insulin just to get medical attention. How do you distinguish between the two? You measure C-peptide. If C-peptide is high, that means that insulin that is in the body, your body is releasing it. That means it's probably a tumor of insulin, insulinoma. If the C-peptide is low, that means the guy is administering insulin to himself. Does this slide make sense? Insulin synthesis and the C-peptide business. Does all of this make sense? Okay. This is just showing you the structure of insulin. There is an A chain, there is a B chain, two disulfide bridges between them, one disulfide bridge within the A chain. So one intra-chain disulfide bridge, two interchain disulfide bridges. This is showing you the synthesis of insulin. Insulin is synthesized with a signal sequence. So this is pre-pro-insulin. The signal sequence is removed. Signal sequence is removed to form pro-insulin. This is the insulin part. This is the pro part, which is then this pro part gets cleaved. So now you have insulin and C-peptide. C-peptide and insulin are stored within the same vesicles, and they're released together. By measuring C-peptide, you have an idea of how much insulin the body is releasing. That's important because it helps you distinguish hypoglycemia secondary to an insulin secreting tumor, or hypoglycemia secondary to exogenous insulin administration. I hope that makes sense. Okay, so mechanism of insulin secretion. I told you to remember Glut2 and glucokinase. What did I tell you to remember about these two? About Glut2 and glucokinase. What do you remember? Glut2 has high Km, and so does glucokinase. I told you that glucokinase is just like Glut2, right? Why is this important? Because these Glut2 and glucokinase are present in the liver, obviously, but they're also present in the pancreatic beta cells. Why is this important? Because when you eat a high carbohydrate meal, a lot of glucose now goes into the pancreas because of Glut2, and a lot of glucose is trapped within the pancreas because of glucokinase. Because of both of these properties, glucose uptake increases. And when glucose uptake increases, you have more glycolysis and you have more ATP production. When you have more ATP production, that closes ATP-sensitive potassium channels. When ATP-sensitive potassium channels close, that causes depolarization of the cell because potassium can't move out of the cell anymore. So potassium stays in, more positive charge inside the cell gets depolarized. This causes opening of voltage-gated calcium channels, voltage-gated calcium channels. Calcium comes into the cell, and insulin, the vesicles containing insulin and C-peptide are released from the cell, okay? Now, this is just a diagrammatic representation. Glucose is high inside the blood because you've just eaten a meal. It comes in to the pancreatic beta cells because of Glut2, which increases uptake of glucose proportional to substrate concentration. Glucose is phosphorylated by glucokinase from glucose 6-phosphate, and this also has a high Km value. Then it generates ATP. High ATP because of more glucose coming in, this closes ATP-sensitive potassium channels. This more potassium stays inside the cell. More potassium staying inside the cell causes depolarization, which opens calcium channels. Calcium comes into the cell. Vesicles containing insulin and C-peptide are released from the cell. It's important for you to know that sulfonylureas are drugs, okay? Sulfonylureas are drugs used to treat type 2 diabetes. In late stage type 2 diabetes, your beta cells get exhausted from producing insulin. And if you give them sulfonylureas, this sulfonylurea will close the ATP-dependent potassium channel. So the sulfonylurea will cause depolarization and release of insulin. Does this make sense? So sulfonylurea is basically mimicking the normal physiology. It's inhibiting the ATP-dependent potassium channel without all of this pathway happening. Okay? Okay. Now, it's important for you to, obviously, you know that high blood glucose causes insulin release, but it's important for you to know that amino acids also cause insulin release. And we'll get to that later on. The third important point on this slide, the first one was sulfonylurea. The first one was the mechanism and the mechanism of sulfonylureas. You should remember that. The second one was amino acids stimulate glucose release, insulin release. And the third one is GLP-1. Basically, GLP-1 is released from the small intestine. When you've eaten a meal, GLP-1 is released from the small intestine and it stimulates insulin secretion. So now, does it make sense to you that if you give glucose IV versus if you give glucose orally, the oral glucose will cause the better stimulation of insulin because of GLP-1. If you give glucose IV, GLP-1 won't be released. So therefore, that extra stimulus for insulin secretion is not there. And so you have less insulin release. If you administer glucose orally, glucose goes up and amino acids as well, but in addition, you have GLP-1 release. So this causes insulin secretion in response to a meal. Therefore, insulin secretion, insulin release after orally administered glucose is higher compared to if that glucose was given IV. Does this make sense? Yep. Okay. Obviously, low blood glucose will stimulate glucagon, not insulin. So low blood glucose will inhibit insulin. That should make sense. Secondly, physiologic stress, because physiologic stress stimulates the sympathetic nervous system, and that causes acetyl-CoA release, not acetyl-CoA, epinephrine release. Epinephrine, as I told you, is a counter-regulatory hormone. It increases blood glucose and it increases glucagon release. So it will inhibit insulin. Okay, so low blood glucose and physiologic stress because of epinephrine, these inhibit insulin release. Because of both of these scenarios, stimulate glycogen breakdown, production of new glucose by gluconeogenesis, and fatty acid breakdown. Does all of this slide make sense? Okay, good. So remember this pathway, remember sulfonylureas, remember amino acids, GLP-1, and this is quite common sense, really. So I don't think you need to focus on this one. Now we're done with insulin. Let's move on to glucagon. Glucagon is just this one slide. It's released from alpha cells of the islets of Langerhans of the pancreas. It is stimulated by low blood glucose, and it causes increased blood glucose by acting on the liver. We focused on this. Epinephrine acts on skeletal muscle and adipose tissue. Glucagon acts on the liver. Stimulus is low blood glucose. Another stimulus is catecholamines because of physiologic stress. I told you, right? Physiologic stress increases glucagon release, inhibits insulin. This is also important for example, let me give you guys an example. Type 1 diabetes, there's lack of insulin. And type 1 diabetes patients are prone to developing ketoacidosis. And they usually develop ketoacidosis after an infection, because that infection causes physiologic stress. That physiologic stress causes catecholamine release. That catecholamine release increases glucagon, decreases insulin. So that further increases lipolysis. And this further increase in lipolysis causes excess acetyl-CoA, which encourages ketone body production on a high level. So this is why this stimuli is important, physiologic stress. That's why I'm emphasizing on this, because we'll discuss it later on. Secondly, also, if you remember from the last slide, I told you amino acids stimulate insulin, but they also stimulate glucagon. That means if you eat a high protein meal, that will stimulate both insulin and glucagon, and so your blood glucose will stay normal. So a high protein meal doesn't alter blood glucose because it stimulates both insulin and glucagon. If it stimulated one or the other, then blood glucose would change, but it doesn't because it stimulates both. Does this slide make sense? It's quite minor in my opinion. Yeah. Okay. This is the last slide. This is the second last slide of the second lecture. Hypoglycemia. Hypoglycemia is defined as a blood glucose level less than 70. There's two physiologic responses, as it should make sense to you. Glucagon release, obviously. Second, the hypothalamus triggers release of catecholamines, epinephrine, norepinephrine, growth hormone releasing hormone, and ACTH. ACTH stimulates cortisol release. Growth hormone releasing hormone stimulates growth hormone release. All of these are the counter-regulatory hormones. So hypoglycemia causes glucagon release, epinephrine, cortisol, and growth hormone. Okay, the glucagon and epinephrine have immediate effects because they act by phosphorylation, dephosphorylation. Glucagon activates glycogen's breakdown by dephosphorylating glycogen phosphorylase. Epinephrine acts on lipolysis. Cortisol, if you remember, is a steroid hormone. Steroid hormones have intracellular receptors. So cortisol goes inside the cell because it's lipid soluble. It goes inside the cell and its receptor is inactive. Okay, this receptor, cortisol, as soon as it binds to its receptor, the receptor opens up. And the receptor has certain fingers. We call them zinc fingers. This cortisol receptor with zinc finger, this complex goes to the nucleus and binds to areas called response elements. This causes increased gene expression of proteins that stimulate gluconeogenesis. But as you know, gluconeogenesis takes time. So the effects of cortisol and growth hormone, etc., are long-term because they're affecting gene expression. Okay. Now, if any of you attended my cardiac function curve PAL session, you will remember that I told you, or probably you don't remember, that I told you that alcoholics are susceptible to developing hypoglycemia. But how do you understand the mechanism? It's very, very easy. Look, the liver cell has a limited supply of NAD, of NADH, and it's utilizing NAD in a lot of different pathways. NAD is utilized in glycolysis, is utilized in the Krebs cycle and in oxidative phosphorylation, is utilized in lactic acid production, is utilized in is utilized in beta oxidation, right? So it's utilized in many, many different pathways, but the supply of NAD is limited. That means the liver cell needs to generate NADH and quickly recycle the NAD for it to be used in other pathways. Does this make sense so far? Or should I repeat this one more time? NAD is in a limited supply in the liver, so the liver needs to regenerate NAD very quickly. So what happens? Ethanol is alcohol, right? Ethanol gets converted to acetate and it generates 2 NADH. And the liver, and if you've consumed a lot of alcohol, like alcoholics do, you've generated a lot of NADH. Now the liver cell is a bit worried because it needs to regenerate NAD, right? So what it does is it converts pyruvate to lactate, and that regenerates the NAD, and it converts oxaloacetate to malate. You don't need to remember these steps specifically, okay? But I'm telling you this because pyruvate and oxaloacetate are used in gluconeogenesis. So by converting pyruvate to lactate and oxaloacetate to malate, these can no longer go into gluconeogenesis. And so alcoholism will decrease gluconeogenesis because of this increased NADH production. And alcoholics are unhealthy, as you know, they don't eat for long periods of times. So their body is carrying out gluconeogenesis. But if they drink a lot of alcohol, this will prevent gluconeogenesis. So they will get hypoglycemia. Does this make sense? Good. Let me ask you a question which will, it's not in MOL 2 course, but it's interesting to know. Suppose an alcoholic comes to you with hypoglycemia. You think, okay, I'm just going to give the guy glucose. And the guy dies because of lactic acidosis. That's the scenario I'm giving you guys. The guy comes to you, an alcoholic comes to you with hypoglycemia. You say, okay, I'm just going to give the guy glucose. And the guy dies because of lactic acidosis. Why? Why did the guy die because of lactic acidosis? Does anyone know? Whoever knows the answer to this, I'm going to give them 5 reals. Just to motivate you guys. It's very interesting because alcoholics, do you know what other nutritional deficiency alcoholics are susceptible to? Any vitamin deficiencies that come to mind? Anything? Alcoholics can develop thiamine deficiency. If alcoholics develop, alcoholics can develop thiamine deficiency. If you remember, pyruvate goes into acetyl-CoA production by an enzyme called pyruvate dehydrogenase, which requires thiamine. Alcoholics are usually thiamine deficient. So alcoholics can't convert pyruvate to acetyl-CoA. So if you give the guy glucose at that time, all of the pyruvate that they will generate, it tries to go into acetyl-CoA production, but it can't because there's no thiamine for PDH to work. So all of that pyruvate goes into lactic acid production. So the guy will get lactic acidosis. So whenever in an alcoholic, if you get an alcoholic presenting to you with hypoglycemia, which is common in the West, you should always give the guy thiamine first, and you'll learn about this later on. You should always give the guy thiamine. Okay? So just for your information. So I hope this makes sense. Now we're on to the last lecture, diabetes. Diabetes, I will define diabetes. This is a definition that works for me as a lack of insulin effect resulting in hyperglycemia. I was quite specific with the words lack of insulin effect. Lack of insulin effect can be due to 2 things. Either you don't have the insulin, like in type 1 diabetes, the beta cells are destroyed by an autoimmune disease. So you don't have the insulin. So that causes lack of insulin effect. Or in type 2 diabetes, you have insulin, but the cells aren't responding to it. That also gives you lack of insulin effect. But it's important to know that in type 1 diabetes, there's an absolute insulin deficiency, as in you don't have insulin at all, versus in type 2 diabetes, because there's a lot of insulin, you still have some residual or low effects of insulin. So you have a relative insulin deficiency in type 2, whereas an absolute deficiency of insulin in type 1. I put the sentence here for you to recall the biochemistry behind hyperglycemia. We discussed that diabetics can't uptake glucose into skeletal muscle and adipose tissue because GLUT4 isn't there. We said glucokinase isn't working in the liver. So glucose comes into the cell, it's not phosphorylated, it goes back out into the cell. We said the liver is not producing glycogen, the liver is not producing fatty acids. And because you have so you have, it's a balance, right? You have insulin and then you have glucagon. If you take away the insulin, you only have the glucagon now. So if you have only glucagon, the liver starts to synthesize glucose by glycogen breakdown and gluconeogenesis. So that was the biochemistry for hyperglycemia. I hope you all remember that. I saw in your lecture that this was one of the lectures which could come up in your SAQ. I feel like this could be what they ask you about in your SAQ. The diagnosis of diabetes. This is important for you to know. HbA1c is glycated hemoglobin which is bound to glucose. If it's greater than 6.5, is diabetes. If you give the guy glucose and you measure his blood glucose 2 hours later and it's above 200, that's diabetes. Or if you have the guy fast overnight and then take his blood glucose and it comes up as greater than 126, this is also diabetes. Remember these three criteria, okay? HbA1c greater than 6.5, 2-hour oral glucose tolerance test. Basically, you give the guy glucose and measure it after 2 hours. If this is greater than 200, this is diabetes. Fasting blood glucose greater than 126 is diabetes. Now, let me give you some more information. Normal HbA1c is less than 5.7. Normal HbA1c is less than 5.7. Normal 2 hours OGTT, oral glucose tolerance test, is less than 140. Normal fasting blood glucose is less than 100. Okay, so 5.7 versus if it's less than 5.7 is normal, greater than 6.5 is diabetes. Less than 140 normal, greater than 200 diabetes. Fasting blood glucose less than 100 normal, more than 126 diabetes. All the values in the middle, so 5.7 to 6.5, 140 to 200, 100 to 126 is called pre-diabetes. Okay, so you have to know these values. Now, it's important for you to know that if you measure this OGTT or FPG fasting blood glucose, this will just tell you about the glucose level in the diabetic at this moment. It won't tell you how well the glucose is controlled over time. Whereas HbA1c is an indicator of long-term glycemic control because glucose binds hemoglobin and hemoglobin lasts as long as the red blood cell lasts, right? Which is 120 days. So HbA1c can give you an indication of long-term glycemic control. So remember this. I feel like they could ask you this in your SAQ. The diagnosis of diabetes with these values plus HbA1c is an indicator of long-term glycemic control. Is this slide clear? Yep. Okay, good. What's normal FBG? Normal FBG less than 100. Normal OGTT less than 140. Normal HbA1c less than 5.7. Anything between diabetes and anything between normal and diabetes is called pre-diabetes. Okay. No, fasting blood glucose. Okay, this is FBG is fasting blood glucose. This is how I like to think about diseases in general. Okay, regardless of whether it's diabetes or any other disease, I like to define it first. Then I need to think about the epidemiology of the disease. Do you know what epidemiology means? You guys know what epidemiology means? This is the last slide, by the way. What does epidemiology mean? Yeah, okay. So epidemiology basically means who gets the disease. Is it more common in males or females? Is it more common in elderly people or younger people? Is it more common in obese patients or normal weight patients? Is it more common in hypertensive patients or people who don't have hypertension? Is it more common in America or in the East, for example? So epidemiology is basically to do with risk factors for the disease. Okay, who gets the disease? The pathogenesis is how the disease developed, the mechanisms behind it. Then I like to discuss the clinical findings. Then the lab diagnosis. then the treatment, and then any complications. So this is a method which I use to sort of organize diseases. And hopefully this will make it easier for you. How I defined diabetes in the previous slide was what? Lack of insulin effect. That insulin effect, that lack of effect can be due to lack of insulin, as in type 1, or resistance to insulin, as in type 2. So that's the definition done. Then let's move on to epidemiology. Who gets type 1 diabetes? Can anyone answer that? Who gets type 1 diabetes usually? If you see a type 1 diabetic, who is that guy usually? An infant, yeah. So most of the cases of type 1 diabetes are diagnosed less than 18 years of age. Contrast this with type 2, where most of the cases of diabetes are diagnosed in elderly people or middle-aged to older age people. Okay. Type 1 diabetes is an autoimmune disease, and there's a general principle that one autoimmune disease is associated with other autoimmune diseases. So type 1 diabetes presents in younger people. They are not usually that obese, and they might have other autoimmune diseases. Type 2 diabetes presents in middle age or older age. These patients are usually obese. That's very important to know. These patients are usually obese or have a very inactive lifestyle. What's the mechanism behind this? Refer to your cell signalings lecture. In your cell signalings lecture, I remember this because we did this in MOL 1, that in obese patients, you have an elevated amount of free fatty acids in the circulation. These elevated free fatty acids in the circulation interfere with the signaling pathway of the insulin receptor. So this way, obese patients can develop insulin resistance because the fatty acids are interfering with the signaling cascade of the insulin receptor. Okay, so epidemiology, know that type 1 develops in younger patients, usually normal weight and associated with other autoimmune diseases. Type 2 diabetes, older age patients, obese patients. The third point you need to know about epidemiology is that there's a strong genetic predisposition to type 2. It's stronger than type 1. So if you have a first degree relative who has type 2, you are more likely to develop type 2 diabetes compared to if that guy was type 1 diabetic. So the genetic predisposition for type 2 diabetes is stronger. The pathogenesis, the type 1 diabetes results in destruction of beta cells, resulting in insulin deficiency, in absolute insulin deficiency, and abnormal glucose control. We discussed the biochemistry. Increasing resistance to insulin. So the insulin is there, but the cells aren't responding to it. Initially, what happens is beta cells think, oh, insulin, we need to secrete more insulin. So there's a lot of secretion of insulin. With time, the beta cells get exhausted and you have decreased insulin. Okay, so that's the pathogenesis. Then let's discuss clinical findings. The main problem in diabetes was hyperglycemia. So the problem with hyperglycemia is that glucose can spill over into the urine to give you glucoseuria, which gives you polyuria. The guy's urinating a lot. If the guy's urinating a lot, he's losing a lot of water. So he needs to hydrate himself. So you have polydipsia, increased thirst. Also remember that in diabetes, you have unopposed effect of epinephrine, which causes fat breakdown and muscle protein breakdown. And this causes weight loss. So you have unexplained weight loss, fatigue, because glucose, even though it's in the blood and you have hyperglycemia, glucose is not entering cells because there is no insulin. So you have fatigue of muscle cells, okay? Blurred vision. To understand blurred vision, do you know what pericytes are? Pericytes. Do you know what pericytes are? So if you have capillaries, around those capillaries, there are cells called pericytes, and they basically prevent the capillary from being leaky. In type 1 diabetes, what happens is glucose accumulates. In diabetes in general, glucose accumulates in those pericytes and causes their dysfunction. So the capillaries in the eye become leaky, and you have leakage of blood into the retina, which causes blurring of vision. Okay. The same with type 2 diabetes. So polyuria, polydipsia, polyphagia, fatigue, blurred vision. So you can see the clinical presentation of type 1 and type 2 is largely the same. So how do you distinguish them clinically? Let me ask you. If a guy presents to you with polyuria, polydipsia, fatigue, blurred vision, how do you know the guy has type 1 or type 2? And you diagnose him with HbA1c, and other lab tests. How do you know it's type 1 or type 2? You know, because of epidemiology, right? So if the guy is presenting to you is a young patient, normal weight, other autoimmune diseases, you would think about type 1. If the guy is an obese patient, middle-aged, as family members with type 2 diabetes, you would think about type 2. So that's where epidemiology becomes important. Okay, so we've discussed definition, lack of insulin effect, epidemiology we've discussed, pathogenesis we've discussed, clinical findings we've discussed, lab diagnosis, HbA1c, OGTT, and FPG, 6.5, 200, 126. Normal is 5.7, 140, and 100. Anything in between, pre-diabetes. Then treatment. The main problem in type 1 diabetes is you don't have insulin. So you can just give insulin. That's the mainstay of treatment of type 1 diabetes, insulin. You give insulin, okay? Type 2 diabetes, you have insulin, but the cells aren't responding to it. So there's a drug called metformin, and there's a drug called thiazolidinediones or glitazones. These basically increase insulin receptor sensitivity. And so they counteract this insulin resistance. You don't need to know these drugs, I don't think, okay? So then you have complications. So treatment, type 1 diabetes, insulin. Type 2, drugs which increase insulin sensitivity. I told you at later stages of type 2 diabetes, the beta cells can get exhausted, so insulin secretion decreases. In this situation, you can give insulin, or you can give agents which stimulate insulin, like sulfonylureas we discussed, right? The sulfonylureas act and inhibit the potassium channels, and they cause insulin secretion. So we can give sulfonylureas in type 2 diabetes. Then we move on to complications. Complications, you can divide them into acute and chronic. Acute complications for type 1 is ketoacidosis. For type 2, we said there's still some insulin effect, and so this usually prevents ketoacidosis. The complication of type 2 is called hyperglycemic, hyperosmolar, non-ketotic syndrome. Hyperglycemic, they have hyperglycemia. This hyperglycemia increases serum osmolality, which drags water outside of cells. Then you have non-ketotic, because they have some insulin, they don't develop ketosis. And so this causes a syndrome called hyperglycemic hyperosmolar non-ketotic syndrome, or HHS, hyperglycemic hyperosmolar syndrome or state. So type 1 diabetes complication is DKA. Type 2 diabetes complication is HHS, okay? Chronic complications is what we discussed at the beginning of the lecture. The fact that glucose likes to bind proteins, and that causes ischemia of the foot, leading to amputations. That can happen in the coronary arteries, giving you MI. That can happen in the cerebral arteries, giving you stroke. That can happen in the retinal arteries, giving you blindness. So these are chronic complications of diabetes, okay? Now, definition again. So let me just repeat this one more time. Definition, lack of insulin effect because of two situations. Either you don't have insulin, or the insulin is there, you're not responding to it. Epidemiology, type 1, young patient, normal weight, autoimmune diseases, not that much family history. Second, epidemiology of type 2, obese patient, middle-aged with a family history. Pathogenesis, type 1 is autoimmune mediated. Type 2 is insulin resistance because of obesity. Clinical findings are the same: polyuria, polydipsia, polyphagia, blurred vision, fatigue, weight loss, these sorts of things. Lab diagnosis, HbA1c, OGTT, FBG. Treatment, type 1 is insulin. Type 2 is drugs that increase insulin sensitivity, like metformin. Later stages, you can use sulfonylureas, for example, to stimulate insulin secretion. Complications are acute. In type 1, you have DKA because you have absolute insulin deficiency. In type 2, you don't have DKA because you have relative insulin deficiency. The complication of type 2 is called HHS. Chronic complications because of glycosylation of proteins. That causes ischemia of the foot, retinopathy, stroke, MI, blindness, these sorts of things. Okay, does diabetes make sense to you? The biochemistry I'm not covering right now because we've already covered it in great detail. So we don't need to cover it again. Okay, yeah. So this is just showing you the pathogenesis of DKA. In type 1 diabetes, you have absolute insulin deficiency. Counter-regulatory hormones like glucagon, epinephrine, cortisol are increased. The lack of insulin and the increase of these hormones drives lipolysis. Free fatty acids, lipolysis causes more fatty acids to go to the liver, generating more acetyl-CoA, which is shunted into ketone body production. Ketone bodies are acidic, so they cause acidosis. Free fatty acids in the bloodstream because of lipolysis also cause hyperlipidemia. Absolute insulin deficiency and increased counter-regulatory hormones decrease glucose utilization, increase glycogenolysis, and increase gluconeogenesis, all of which give you hyperglycemia. The hyperglycemia leads to glucose ending up in urine, giving you polyuria. Because of this, you have dehydration, giving you polydipsia. And that's the pathogenesis of type 1 DKA. In type 2 diabetes, you have a relative insulin deficiency. So you don't really have abnormal you don't really have ketogenesis. But the same thing applies. You have increased glycogenolysis, increased lipolysis, increased protein breakdown. So increased gluconeogenesis, which gives you hyperglycemia. Because of this hyperglycemia, water is coming out of cells. And in the brain, for example, if that happens, that can put you into a coma. So that's why you need to treat this immediately. Okay. What type of patients get DKA? Because you have absolute insulin deficiency, if the guy with type 1 diabetes is taking insulin, he's unlikely to get DKA. So the classic patient you will see getting DKA is a patient who is type 1 diabetic, but he's poorly controlled. He's not taking his medication. He's not taking insulin. So that is causing the absolute insulin deficiency. The other type of patient who can get DKA is a type 1 diabetic who doesn't know he has type 1 diabetes. So he doesn't take insulin, and this causes absolute insulin deficiency, and he presents to the clinic with DKA. And then when you resolve the DKA, you find out he has type 1 diabetes. The other situation is a guy, type 1 diabetic, who develops an infection or who develops some sort of stress, because I told you that stress activates epinephrine and glucagon and inhibits insulin. So this drives DKA. So type 1 diabetic who's poorly controlled or who has an infection, or upon presentation, he doesn't know he has diabetes. Okay. And that's the whole lecture done. We finished like a few lectures in less than 2 hours. Any questions? Thank you. Okay. Thank you, everyone. I think we can stop the recording now.